The main reason why we perform in ovarian tissue cryopreservation is fertility preservation, cancer treatment. It's very important that we can protect the fertility. And we have two possibilities. The first one is opening the spot. First, it's quite well known, and the second possibility is vitrification. And the main advantage, which is from the IVF lab system, vitrification avoids ice freeze information when applied properly. So that's the major advantage. So when we compare the situation to the IVF labs, you can see a shift around the year 2000 from slow freezing to vitrification. And that was the main ratio why the ESHRE guideline says currently vitrification is recommended in the IVF lab. With ovarian tissue, it's a little bit different. So slow freezing is still regarded as the standard approach, but there's ongoing research also around the year 2000. And we have published births from Professor Nao Suzuki in Japan, and then later 2 births published by Professor Sherman Silver in the United States. And we have published our birth that was the first one in Europe. The publication was led by our department head, Professor Sänger, in 2024, and there's another publication from Victoria Keros in Sweden from 2024. And the guideline says, but the guideline is still from 2020, that slow freezing is the standard approach, but vitrification is a promising alternate technique. Okay, so the main difference regarding ovarian tissue to cells is that we have multiple types of cells at different stages. We have oocytes, granulosa cells, and other cells. So it's a little bit more complex, and of course you have to regard highest quality and C certification. And so here you can see an intersection of an ovary where you have in the middle, you have the medullary region where you have advanced stages, and in the outer region, that's called the cortex region, we have the vast majority of the female follicular reserve. And this is our main target when we are performing cryopreservation of ovarian tissue. And the medulla is discarded and the cortex is cryopreserved. So you can see here in the figure on the left an unprocessed ovary. Then we have in figure 4 the cortex tissue prepared for cryopreservation. And in figure 5 you can see the setting for vitrification. And figure 6 is the well-known setting for slow freezing. Of course, it's important that we have some kind of quality measurements. That means we have biopsy punches, we digest with collagenase that activates follicles, and you can measure the follicular viability. That's very important. We do that before cryopreservation and after thawing or rapid warming to estimate how many tissue pieces will be transplanted back. So that's very important. And so you can evaluate, is your process working, is everything fine, and so on. The disadvantage is, of course, with this method, you need a fluorescence microscope. Those are very expensive. And other very good possibilities published from the Christensen group in Copenhagen. So you can use neutral-red. This will also stain follicles reddish, and you do not need a fluorescence microscope. Okay, so I guess everybody, well, all of us, we've heard a lot of about cryopreservation protocols, so we'll keep this short. We have intracellular and extracellular cryoprotective agents. So the first ones are mainly ethylene glycol, propanediol, and so on. And the second groups are mainly sugars. Same with cryopreservation protocols of cells. And it's very important to state that also the slow freezing process is not one protocol. There are many, many slow freezing protocols all over the world. And that's very important. So when we talk about slow freezing, we're talking about many different protocols, and the same is with thawing. There are also many different protocols. Okay, so currently the births published by Professor Suzuki, they're using an increasing ethylene glycol concentration, and that's nearly the same system we are using on the right side. And yes, the carriers are a little bit different, but we nearly have the same concentrations. And so you see, we're also using 10% ethylene glycol, 20, and then 35%, but the incubation time is a little bit shorter, but we're getting out the same results. So that's very interesting for us. And regarding rapid warming, it's very similar than with cells. Also, we have nearly the same approach than the Japanese group. So initially a very high concentration of sugar at 37 centidegree. That is very important to avoid the reformation of ice crystals. Okay, so we did not wake up in Bonn one morning and said, okay, let's start with vitrification of ovarian tissue. That means we did a lot of proper research before. That was the first publication with split samples, slow frozen, vitrified. And then we compared the different follicular viability, and we saw there was no difference. So that was quite encouraging for us. And then we had a second publication because the tissue will be later transplanted. So it's also very important to measure the angiopoietic capacity of the tissue. We did that as well, also with split samples. And we also saw that there is no significant difference. Okay, this is a little bit messed up. I'm sorry. So the main advantage of vitrification is that we avoid ice crystal formation, and we do not need a lot of costly equipment, to place it in a nutshell. Okay, so there's very good research published from Lodovico Parmigiani comparing different kits when comparing vitrification and rapid warming of cells. So the question is, can you use different kits? And this was a question we asked ourselves. Is it possible, for example, that you can apply rapid warming approaching procedures to slow frozen ovarian tissue? And we did another study that was published recently, and we also did split samples. So that means we had slow frozen samples that were conventionally thawed and rapidly warmed, and we also saw that there is no significant difference. So this would be a possibility to unify the protocols, especially when you're getting tissue from another center. You have to use for a transplantation. You do not have maybe the same media, so you can use a universal approach, as Lodovico could show that it is possible with cells. Okay, so summing it up, slow freezing of ovarian tissue is performed by around about 95% of the centers. I would say currently there are nearly 300 births published, but we do not know the exact numbers. And regarding vitrification, is performed by a minority of the centers, around about 5%. And we have currently 6 births published. I talked with now Suzuki, they have about 10 more births, but those are not published. So we estimate currently that there are 15 births after vitrification, rapid warming, and transplantation. And regarding PubMed, so there are 3 major publications, including around about 50 studies, stating that vitrification is at least equivalent to slow freezing and to a certain extent superior. So that's very positive. And we think that we should see both methods complementary rather than competing. But it's also important that we have global initiatives to work on the protocols. That's very important. And also for C certification, media, and so on. Okay. Thanks a lot.